Hello guys, I am Harun and this is my YouTube channel Harun Biozone. Today in this video I will discuss 35 important MCQ questions from the chapter genetics. These questions are very important for any medical examination and also other biological based examination. So don't be late, let's go and start the video. Here the first MCQ question of this video is: if Mendel had chosen to study traits determined by linked gene, what is linked gene? He would not have discovered. If the if Mendel's study the linkage gene or linked gene, he would not discover law of segregation, law of dominance, law of independent assortment, law of purity of gametes. So first, what is link gene? What is linked gene? Linked gene means when the genes present are very closely to the same chromosome. Okay, suppose this is chromosome and the linked genes are present. Suppose A and B are two genes. These genes are present on the same chromosome. So these chromosomes, these genes are unable to separate or show recombination during crossing over. Okay, this is called linked genes. So here law of what is law of segregation? Law of segregation at the time of gamete formation. So, like A, A, B, B. At the time of gamete formation, at the time of gamete formation, gamete formation, the genes are separate from each other. This is called law of segregation. Law of dominance. Law of dominance means suppose if we cross in tall plant and dwarf plant in F1 generation, only tall plant are produced. So here tall is the dominant character. It's called law of dominance character. So the trait that are expressed in F1 generation, that is called law of dominance. Law of independent assortment. At the At the time of gamete formation, the gametes are not only separate from each other, but also they independently mixed up with each other. Okay, this is called law of independent assortment. And law of purity of gametes. Now the gamete purity of gametes means here the alleles are do not mixed up. Okay, this is actually linkage, complete linkage, where no variation occur, only where no variation occur, only parental type gametes are produced. So here the right answer is law of independent assortment. What actually they say, genus that are on the same genes that are present on the same chromosome show linkage. Genes that are present on same chromosome show linkage. But when genes are present on different chromosome, when genes are present on different chromosome, they show recombination. This is they show recombination, but they show linkage. Okay. If two genes show linkage, then two genes do not assist independently, so law of independent assortment would not discovered. If Mendel study the linkage, then Mendel do not provide this formula law of independent assortment. Law of independent assortment is applicable in dihybrid cross. Dihybrid cross. So Mendel do not study or do not provide any information about linkage. Next question is, here some linkage. When the genes are present on the same chromosome, these show linked gene. But when the genes are present on different chromosomes, these are not linkage, they show recombination or crossing over. Okay, and these genes, but here the distance between these two genes are very close, okay, very low, but here the distance between two genes are large. So when the genes present in close together, they show linkage, but if the genes present on only one chromosome, but they show distance between a big distance between them, they do not show linkage. Next question: in which of the following the genotypic ratio are same? In case of incomplete dominance, genotypic and phenotypic ratio are same. Suppose here the red flower and white flower, here RR red flower and RR white flower, then recombination produce RR. This is pink flower. So here the neither R is expressed or nor small r is expressed. Here new characters are formed. This is called incomplete dominance. These characteristics are found in evening prime rose plant. Okay. So here phenotypic and genotypic ratio. Suppose if self cross between RR, then produce RR, capital R, capital R, capital R is small r, capital R is small r, and small r is small r. Here pure red character, then heterozygous red RR character, and homozygous recessive red RR character. So here genotype is 1 is to 2 is to 1, and also here 3 characters are produced, that is phenotype is also 1 is to 2 is to 1. So phenotype and genotype ratio are same in case of incomplete dominance, but in case of codominance like MN blood group, where MN blood group or AB blood group, where M and N both genes are expressed. When both genes are expressed, this is called codominance. Polygenic inheritance. Polygenic means poly means many and genic means gene. When many genes express a single character like human height, human health, etc. Example of polygenic inheritance. Epistasis means when one gene masks the expression of another gene. Say for example, suppose A and B. The A is the tall plant and B is the dwarf plant. Say for example, if they produce, if B suppress the expression of A and produce dwarf character, means here B allele suppress the expression of A allele, this is called epistasis. When one gene masks the expression of another gene is called epistasis. So here A gene is epistatic A gene whose expression is suppressed. So A gene is the epistatic gene, hypostatic gene whose expression is masked, and B is the epistatic gene. B gene suppress the expression of A, so B is in the epistatic gene and A is the hypostatic gene, and this process is called epistasis. So here the right answer is incomplete dominance, where genotype and phenotypic ratio are same. Next question: match the columns and find out the correct combination. Here RR and RR, it is a monohybrid cross, but it is dihybrid ratio. So RR is the matching of RR matching of 3 4 A 4. and RR, heterozygous RR and homozygous RR, the expression of this one, and this is expression of 2, and this is the expression of test cross. This is test cross where F1 generation, F1 progeny were crossed the recessive parents. Okay, here self cross occur where F1 progeny are self. Okay, in this case, here the heterozygous F1 progeny, it is also a test cross where F1 progeny means that suppose RR and RR, then produce produce RR. When this RR, F1 progeny RR is crossed with the recessive parent, that is called test cross. Okay? In case of test cross, there is 1:1 ratio are found. How? This is RR smaller, capital R smaller, smaller. Here RR is to RR, 2:2 means 1:1. So here in case of heterozygous RR and homozygous RR, recessive RR cross produce 1:1 ratio. Here the right answer is A. Next question, a tobacco plant heterozygous for a recessive character. Tobacco plant heterozygous for recessive character is self pollinated and 1200 seeds are subsequently germinated. How many seedling would have the parental type? So here, suppose a tobacco plant heterozygous for a recessive character. Heterozygous means this is the recessive character, is self pollinated, produce the following character: TT, TT, capital small t and small t, small t. So here recessive character, this is recessive, heterozygous for recessive character, heterozygous t, capital T small t and capital T small t. Here is recessive, this is the recessive character. In the question says the heterozygous is the recessive character. The total number of capital T small t are 1 by 2 by 4, that is 1 by 2, and total number of seedling are produced 1200, 1200 to 600. So answer is 600. Okay, this is the point, heterozygous for recessive character. Okay? Right answer is 600. Next question, Andalusian fowl, one type of hen, birds. Andalusian fowl have two pure forms, black and white. If black forms is BB and white forms WW are crossed, F1 individuals appear blue colored. Means here they want to say BB is the black and crossed with white, that is WW are crossed, F1 individuals appear blue colored. So in case of F1 BW, that is blue in color. So incomplete dominance are found here. Due to incomplete dominance, which of the following would be an outcome of cross between black and blue form? Black and blue form means this is cross between BB. So BB and BW if crossed, then what happened? BB, BB, BW, BW, that is 50% black and 50% blue in color are produced. Okay, so that is 2 black and 2 blue, 2:2 ratio. If we calculate it 1:1. So D is the right answer. Next question. A colorblind man, genotype is XCY, has a colorblind sister and a normal brother. What is genotype of father and mother? So here this is father and mother, XX and cross XY, mother and father. Then colorblind sister XZ and normal brother XY. So colorblind sister and brother XY. XY is normal. This is the inheritance pattern. What is the genotype of father and mother? So here looks the two X chromosome in case of daughter. The X chromosome of daughter 50% come from father and 50% come from mother. But in case of male, in case of son, the X chromosome is derived from mother. Okay. So this X chromosome, suppose this is, if mother is heterozygous and father is affected, then the daughter is affected. Okay. And 50% chance of the son become not affected. So here the right answer is XCY and XC. Colorblind father and normal mother and carrier mother produce colorblind daughter and normal son. Which of the following disease is an autosomal recessive disorder? In our body, there are two types of chromosomes are present: autosome and allosome or sex chromosome. So autosome is 22 pair and one pair is allosome, total 46 pair. So autosomal recessive, the autosomal chromosome are affected in which disease? First, color blindness. Color blindness is the X-linked recessive disorder. X-linked recessive disorder, where thalassemia is autosomal recessive disorder. Autosomal recessive disorder. Recessive disorder. Here hemophilia is X-linked recessive disorder. Linked recessive disorder and Huntington disease is a inherited disease that cause progressive breakdown of nerve cell. Huntington disease is a genetic disorder, but it break down the nerve cell that affect the movement nerve cell of brain. This is autosomal disorder. Okay, autosomal dominant disorder. In case of when the brain nerve cell is destroyed, the movement, affect, thinking, memory, etc. are affected. So here the right answer is the autosomal recessive disorder is thalassemia. It is, so colorblind is the X-linked recessive disorder, hemophilia is X-linked recessive disorder, Huntington disease is autosomal dominant disorder, and thalassemia is a autosomal recessive disorder. In case of color blindness, there are three types of color blind are produced. In our eyes, in cone cell, there are three type of pigment are formed, that is blue, green and red. Okay, if any one of the color is not produced or pigment not produced, then we do not perceive the that color. This is called color blindness. Okay, thalassemia, in case of thalassemia, here hemoglobin do not produce properly, so anemia, severe anemia are produced in our body. In case of hemophilia, blood clotting factor 8 and 9 do not produce, so blood will continuously flow from our body, from the cutting portion of the body. Okay, next question is, In a plant, gene A is responsible for tallness and is recessive allele A for dwarf. B is responsible for red flower. Here, tall plant and red flower, that is AABB. Okay, and the B flower and color and its recessive allele and red flower color and its recessive allele B for white flower color crossed with AMBB, that is dwarf plant and white flower. A tall and red flower plant with genotype AABB plant, that says tall and red flower plant with genotype AABB crossed with dwarf and red flower AABB. Okay, so here the produce AABB is crossed with AAB and B, that is dwarf dwarf and red flower. Okay, what is the percentage of dwarf white flower offspring? Dwarf white flower is A, B and B. What is the percentage of? Here technique, one technique I will explain. Here A cross with A, okay, then A, this A crossed with A produce A and A. Okay, so here A, what is the percent of small a? That is the percent of small a is 1 by 4 and 1 by 4. Sorry, 1 by 4, 1 by 4. So total number of 2, that is 2 by 4 into, then crossed with B and B and B and B, then produce BB, capital B, small b. So here 1 by 4, 1, here produce 1 by 4, 1 by 4. So here in this case, when A is found, 1 by 4, 2 by 4 into 1 by 4. So here 1 by 2. So 1 by 2 into 1 by 4, that is, or ignore it, where 1 by 4. So 1 by 4, 2 by 4 into 1 by 4, that is 2 by 16. Total number of 2 by 16 into 100, that is 4 and 425. So 2 and 2, 2 and 12.5, that is the right answer. Okay, next question is, read the following and identify the wrong statement. Identify the wrong statement. During Mendel investigation, statistical analysis and mathematical logic were applied to problem of biology for the first time. Not. Mendel is a, maybe Mendel was a mathematician and physicist, but he worked on biology. So he do not apply mathematics or physics on biology. Mendel selected 14 true breeding pea varieties. Mendel selected 14 not pea varieties. Mendel was the first to give experimental verification for the chromosomal theory of inheritance, and Mendel experiment had large sampling size which have greater credibility to the data collected. This is the correct Mendel first time, because before Mendel there is no any scientist are present in this world that work on genetics. Okay, Mendel only the first person who worked on genetics. The next question is, the minimum weight of tomato obtained from a plant is 40 gram. The maximum weight of tomato obtained from is 140 gram. What will be the weight of tomato obtained by crossing plants with genotype AA and BBAB? Okay. So here you can see minimum character is the recessive character, recessive. Okay. And maximum is the maximum is the maximum means the dominant character. Okay, dominant. So here says what will be the weight of tomato? Tomato weight. What is the weight of tomato that produce in F1 generation? If we cross AA BB into AA BB, then in F1 generation produce AA BB. This is hybrid. Okay. So here the recessive character, this is 40, this is 40 gram, and dominant character is 140 gram. So these both characters are mixed here. So 140 plus 40 by 2. Okay, two different character, that is 180 by 2, that is 90 gram. So F1 during F1 generation the progeny is tomato size is 90 gram. It is the correct answer. Next question, identify the wrong statement. Here identify the wrong statement. Which statement is wrong? Identify. The probability of homozygous dwarf progeny formed in Crotest cross is 0. So here the probability of homozygous dwarf, that is capital T, small t, small t, no, capital T, and capital T. It is right answer, right option. Because if we cross TT into TT, there is no TTR formed, no TTR formed, no formed. Okay, the probability of homozygous dwarf formed in test was TTT 0. Homozygous dwarf from TT and TT, there is no TT is formed. Okay, it is also right point. The heterozygous tall progeny formed in cross TT 25%. Heterozygous tall means TT and TT. Not, it is not right, because if we TT cross TT, then we find TT TT capital T, capital T, capital T, T small t, and t small t. So there is 20, not 25%. This is 50%. The answer 50%. So this is the wrong answer. And the homozygous tall progeny formed in a cross TT. If both the parent are homozygous tall, then all the progeny are homozygous tall. Okay. So here the wrong statement is the heterozygous tall progeny formed in a cross TT is 25%. Next question is incorrect statement. Incorrect. What is incorrect statements is are Mendel self-pollinated the F2 plants and found that dwarfness cannot appear in F3 and F4. Mendel, if the self-pollinated plant means this is if self-pollinated, then definitely produce 25% TT. Okay, so this is the incorrect one. Here half T and half T, that is means TT cross into TT. Okay, then produce 2 TT half T, ultimate produce TT, TT and TT, that is 1 by 4 TT, 1 by 4, 1 by 2, that is 2 by 4, Tt means 1 by 2 Tt, and here 1 by 4 Tt, that is 1 by 4 Tt. Recessive traits are seen due to non-functional enzyme. So what is why it is called dominant and recessive? Dominant allele produce functional enzyme, but recessive allele do not produce functional enzyme, so they do not express. Okay, so this is the right option. This is right option. This is right option. Mendelian approach 12. Mendelian approach of using mathematics was not accepted by many of the biologists of his time, despite of the fact that Mendel provided physical proof for the existence of the factor. This is not the right option. Here, dominance depends as such on the gene product, and gene products means functional enzyme. Functional enzyme. Okay, gene product means functional enzyme, and the production of particular phenotype. This enzyme produce particular phenotype from this product. It does on the particular phenotype that we choose to examine in case of more than one phenotype influenced by same gene. But one gene influenced many characters. That is called pleiotropy. 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 In case of phenylketonuria, where single gene produce many characters. Okay, if it is right option. In an individuals, only two alleles can be present. Suppose this is tall. So here tall character is represented by capital T, and this gene is made by two allele. Okay, two one and two allele. Okay. Multiple alleles can be found only when population is multiple allele means many alleles when found when the population means group of organism are studied made. This is right answer. So here this is the incorrect answer here. Next question. Correct statements are alteration of DNA sequence result in the change in the genotype only, not genotype. It also phenotypes on it also alters the phenotype. Okay, this is wrong statement, because if suppose GAG, okay, this is amino acid valine. Suppose example, if the sequence is change GUG, then this is stop codon. Okay, so change the genetic sequence, also change the phenotypic change. The analysis of traits in several of generation of a family is called pedigree. Definitely, pedigree analysis means study the disease that appear, genetical disease that appear or not the from generation from before generation. Mendelian disorder are caused due to alteration in single gene. Mendelian disorder are caused due to alteration of single gene. This is right answer. This is right option. Down syndrome and Turner syndrome are example of chromosomal disorder, because Down syndrome is caused by the duplication of 21 number chromosome. Okay, and Turner syndrome is caused by the X0, that is absence of one X chromosome. Here total chromosome number 45, here total chromosome number 47. Okay, genetic disorder. Here right option is A is the wrong answer, but and B, C, D is the correct option. Next question. Sita and Ram have their first child. Sita and Ram have their first child. Sita knows her blood type is A. So here they want to say Sita. Sita has blood group A. Okay, and but Ram does not know. But Ram does not know his blood group. Okay. Then Ram knows both his mother and father have type B blood, but Ram, but father and mother of Ram and Sita have blood group B. Okay. B. The first child is a boy named Kush. Kush has type O blood. Kush, and their first child blood group O. Okay. Sita and Ram do not understand what happened. Here, Ram's father and mother has blood group B. That is, the genotype is IB or IBI. Okay. But Sita has blood group A. That is IA or IAI. So in what Sita and Ram, son of Sita and Ram is blood group O. How it is possible? If Sita blood group is IAI and Ram blood group is IBI, then possibility of 25% possibility of blood group become here IA, IB, here IB, I, here IA, and here II. 25% chance their offspring is O blood group containing. Clear? Next, here the right answer is B. Sita genotype IAIO and Ram genotype IOIO has whose express the phenotype. So here Ram's blood group IBI, this is okay, when this is not answer IOIO, because father and mother has IBI. If father and mother has, so if father is IBI and mother is IBI, then the son is I0 I0. B is the correct answer. Next, select the correct match. Phenylketonuria. Phenylketonuria is an autosomal disorder. Phenylketonuria is an autosomal Phenylketonuria is an autosomal dominant trait. Okay. Sickle cell anemia autosomal recessive trait. Chromosome number 11. Thalassemia is X-linked. Hemophilia is Y-linked. So here the right option is sickle cell anemia is the genetic autosomal recessive trait. This is the right answer. But here thalassemia is a autosomal recessive disorder. Okay. Hemophilia is X-linked recessive disorder. In case of phenylketonuria, it is genetical disease where one enzyme phenylalanine hydroxylase, suppose this is phenylalanine, phenylalanine convert into tyrosine, okay, by phenylalanine hydroxylase enzyme, PHA. If this enzyme are mutated, then tyrosine do not produce. If tyrosine do not produce, actually what is the function of tyrosine? Tyrosine produce melanin. So when this enzyme is mutated, the phenylalanine do not convert into tyrosine, so melanin is not produced, so our skin pigment are not produced here, and phenylalanine deposited in our in our blood and it excretes through blood. It is black urine. It is also called black urine. Okay, that sickle cell anemia is the right answer. Correct match. It is a autosomal genetic changes, not autosomal dominant order. Next question is, if Mendel had studied the 7 trait, so Mendel studied 7 trait, a 7 trait present on 14 chromosome. Okay, this is if this is one chromosome, the trait are present, suppose A, B, etc. In this type, the 7 traits present in 14 chromosome. But if 7 traits present in 12 chromosome, traits in 12 chromosome, then what happened? In what way would this interpretation have been different? How he would not have discovered the law of independence? Definitely, if 7 traits present on the 12 chromosome means ki some traits present on more than one chromosome, some two or more characters or two or more traits present in single chromosome. That is, so here in this case, here suppose many characters are present on the single gene. Okay, suppose A, B, C, D, in where single character present in the chromosome, where the double character or many character present in chromosome. That means these genes show linkage. That genes show linkage. He would have discovered sex linkage. He could have mapped the chromosome. He could have discovered in blending inheritance. Blending. Here, the correct answer is he would not discover the law of independent assortment, because in law of independent assortments the gametes are separate independently from each other. Okay, but in case of linkage there is no separation of alleles or gene occur. So here this is the reason. If 12 7 trait present on 12 chromosome, they discover linkage. If they discover linkage, then law of independent assortment do not discovered. Okay. The next question is, which of the following statement is incorrect? At the time of gamete formation, gamete formation, homologous chromosome bearing the factor pair segregate in anaphase 2 of meiosis and 2 producing and meiosis 2 and producing 2 daughter cell. Here says that at the time of gamete formation, the chromosome bearing factor becomes separated in anaphase 2 of meiosis 2. Both chromosomes and alleles follow law of segregation. Definitely, according to Mendel's law, at the time of gamete formation, all the alleles that present on the chromosome separate from each other. That is called law of segregation. Both chromosome as well as genes occur in pairs in the somatic. Somatic cell means to a diploid number of cell. Diploid number of cell means here a pair of chromosomes are present. Okay, pair of chromosomes are present. In crisscross inheritance, male passes its gene on crisscross means, suppose grandfather to his daughter, then daughter, grandfather to daughter, daughter to son, son to again daughter. This is called crisscross inheritance. It is the characteristic features of X-linked recessive disorder. To the daughter, while the son gets gene on X chromosome from the female. Okay, this is X-linked recessive. X-linked recessive. So what happened? You see here, this is the chromosome. This is the cell. This cell undergo division. Okay, this is the cell. Here during S phase, the cell number of chromosome become double. Okay, during S phase the number of chromosome, S phase of cell cycle, the number of chromosome become double. Then meiosis on of anaphase on, meiosis on and anaphase on these chromosomes are separated. Okay, these chromosomes are separated, but do not the separate, so two nucleus are formed, but the cytoplasm do not divide. Here these cells are separated from each other. Okay, during meiosis, during anaphase 1, then during meiosis 2, that is anaphase 2, anaphase 2, these chromatids are separate from each other and produce 4 haploid gametes. Okay, these chromatids are separated. So in case of meiosis 1, the chromosomes are separated. In case of meiosis 2, the chromatids are separated. So here the incorrect option is A, because this separation of chromosome occur at the meiosis 1 of anaphase 1. Next question is, a gene having many effects, a gene controlling more than one character. So when one gene express many character, that is called polygenic inheritance. That is called polygenic inheritance. Example of polygenic inheritance is the height and body color of human being. But many genes determine a single character. Okay, this is called where a gene having many. Sorry. This is but when a genes having many effect, a single gene show many effect, that is called pleiotropic. That is called pleiotropic gene. And epistatic gene, when one gene masks the expression of another gene, this is called epistatic gene. And this epistatic gene, when suppose A and B, the A suppress the expression of B. So here is the epistatic gene, B is the hypostatic gene, and this process is called epistasis. And what is complementary, what is supplementary gene? Here complementary means when one gene normal Mendel ratio is 9 is to 9, normal Mendel ratio is 9 is to 3 is to 3 is to 1. But in case of complementary and supplementary gene interaction, that is next sir, the ratio become 9 is to 7 and 9 is to 3 is to 4. 9 is to 3 is to 4. Why? When one gene mask or alter the effects of another gene, example called complementary gene. Complementary genes means when one gene masks the expression of another gene is called complementary gene. The example complementary gene example is white variety, white flower and white flower. When here CCPP, capital C, P and P cross with small c, capital P. This type of arrangement of genes so that the genes present on separate chromosome, different chromosome. Okay, that is called intergenic interaction. Intergenic, or that means these genes are present on separate chromosome. So when these dominant allele present separately, when these dominant allele present separately, they do not express itself, but when they come together, they express and produce this is purple color. Okay, when these dominant genes are present on different chromosomes, they do not express themselves, but when these two dominant genes are come together, they togetherly express new characters. This is called complementary gene. You can see here, this is purple color, both the dominant alleles are present. In case of white, but in case of white, either capital C, small p, or capital C, small p means here C is present, not capital P is present, then only express white in color. So it do not maintain the Mendelian inheritance. It show the genetic ratio, genotypic ratio or phenotypic ratio is 9:7. Only purple and white are produced. In case of complementary gene, in case of supplementary gene interaction, genes that can only be expressed in the presence of other gene. Okay? Here capital A, capital B is the agouti color of mice, here albino color, small albino color. Okay, here dominant, this is recessive, but when these dominant are present, they accumulately they produce agouti color. Okay, agouti color is produced. Here in this case, A represent agouti color, B do not express, B has no expression. Okay, so when the but when A and B present accumulately, they produce a color. Okay, when A and B, one of them are absent, they produce different color. Suppose in case of A B B, means here capital B is absent, so it produces black color. Okay, again if A B B, here capital A absent, only small b is present, that produce black albino color. Okay, when both A and B, capital A, dominant A and B are present together, they produce agouti color, but if absence of any of them, produce different color. Here, here the ratio 9:3:4, complementary and supplementary gene interaction. So right answer is pleiotropic. Right answer is pleiotropic. Next question is, in a if in Drosophila, red and white eye color are sex linked character. Okay? Sex linked means X linked, that means the genes present on X chromosome. In which chromosome are the sex linked characters located? In case of Drosophila, this sex linked character X. So here the chromosomes are present on X chromosome, but in case of human, the Y linked, that is the sex determination region present in the on the Y chromosome. Next question is, percentage of recombination between A B is 9%, A and C is 17%, B and C 26%. Then the arrangement of genes. So according to chromosomal theory of inheritance by Bateson and Punnett, Bateson, genes are linearly arranged. Genes are linearly arranged on chromosome. Okay, chromosome. Genes are linearly arranged on chromosome. So here the distance between A and B. So first you see the maximum distance. Okay, maximum distance. So here between the B and C, the distance is 26%. Okay, and then second maximum is C. B and C and A and C. So suppose here if we set A here, the A and C distance is 17%, and A and B distance is 9%. Okay, so here the right arrangement of the gene is B, A and C, that is D is the correct option. Next question, if the DNA codons are ATG, ATG, ATG and cytosine base is inserted at the beginning, then which of the following will result? Suppose here ATG, T, G and ATG present. Then cytosine is added at the beginning. So if cytosine arranged, the three code separated. Okay, because three codes produce one amino acid GAT then G. Okay, this is the correct sequence. CAT CAT GAT GATG. A is the correct option. Next question is, the color blindness is more likely to occur in males than in females. Here says color blindness is disease, genetic disease that appear more in male than female. Why? First, color blindness. What is color blindness? It is X-linked recessive. X-linked recessive character. Okay. So here, the color blindness is more likely to occur in male because in case of father X, mother XX present and father XY present. Okay. When the gamete is produced, XX and X and Y. Clear? So if Punnett square is occur XY, then what happen? In case of daughter, this is mother and the father region. So mother, one X chromosome from mother and one X chromosome from father. Okay. In case of daughter, one X from father, one X from mother. But in case of son, the Y chromosome coming from the father and X chromosome from mother. So in case of daughter, the X chromosome coming from mother, but in case of son, the Y chromosome coming from father and X chromosome coming from mother. Okay, XY. So then Y chromosome of males have the genes for distinguishing color? Not. It is X linked. That means the effective genes are present on X chromosome. Genes for characters are located on the X chromosome. The trait is dominant in male and recessive in female. The trait is dominant in males and recessive in females. Genes or for characters are located on the X chromosome. B is the correct option. Next question is, a cell at telophase stage is observed by a student in a plant brought from the field. He tells his teacher that this cell is not like other cell at telophase stage. Okay, there is no formation of cell plate, thus the cell is containing more number of chromosomes as compared to other. Suppose here what happened, this is cell. Okay, this cell contain two chromosomes. So say for example, if the after the cell division, the cell plate is formed. Okay, cell plate is formed and two chromosomes are separate, ultimate produce two cell with chromosome. But if the cell plate is not formed, okay, then what happened? The one cell carries both the chromosome and one cell is nothing. Okay, the chromosome number become double. Okay, this is called polyploidy. When the and this process is called non-disjunction. It is a natural process, but it is also occur by artificially when colchicine is a microtubule inhibitor. If colchicine is applied, the microtubule do not produce in cell division, so chromosome do not separated. So one cell carries large number of chromosome but double number of chromosome, but another cell do not. So it is called polyploidy. What is aneuploidy? Aneuploidy means chromosome number. Polyploidy means set of the chromosome. Set of the chromosome changes. Like example 2n, 3n, 4n, 5n, 6n, etc. But in case of aneuploidy, if the chromosome number, normal chromosome number, what is 46? When the chromosome number more than 46, more than 46, that is called hyperploidy. Hyperploidy. Okay. When the chromosome number is less than, is called hypoploidy. Hypoploidy. Clear? Somaclonal variation means somaclonal. Clonal means the formation of parental type of protein. Soma means somatic cell. Okay, autosomal chromosome. Variation. When the variation occur in somatic cell, that is called somaclonal variation. Next question is, pick out the correct statement. Hemophilia is a sex-linked recessive disease. Yes, this is right answer. Down syndrome is due to aneuploidy. Down syndrome means chromosome number increase. That is, the chromosome number become 47. Okay, this is also right answer. Phenylketonuria is an autosomal recessive disorder. Yes, this is the right answer. So B is the correct answer. And sickle cell anemia is an X-linked recessive disorder, not false. Sickle cell anemia is a chromosomal disorder where point mutation occur. How it is occur? In case of sickle cell, a normal case, RBC is a biconcave shape in structure, but in sickle cell anemia, the RBC become sickle-like structure. Why? Because here one amino acid is glutamine converted into valine by a single point mutation. Suppose GAG. In case of DNA level, TTC. Okay. This is here point mutation occur in A and convert into G, convert into GTG. GTG, that is CAC. But when transcription occur, that is RNA level, then produce GAG, but here produce GUG. The protein level, in case of protein level, after translation, this is produced glutamine and this is produced valine. Okay. If this point mutation occur, then the shape of the RBC is changes. Next question is, in the following human pedigree, the field symbol represent the affected individuals. Identify the type of given pedigree. Here, first you can identify it is recessive or dominant. Recessive or dominant. In case of recessive, the generation are skipped. Here, first generation, second generation, third generation, and fourth generation. Okay. The first, here you can see in each and every generation, in each and every generation, someone is affected. Here, first one affected, this they affected, then in next generation, they are affected, next generation they also affected. So first it is a dominant disease. First it is a dominant disease. First point. When the do not skip generation, it is called dominant disease. Then it is autosomal or X-linked. In case of X-linked, the mostly females are affected, but in case of autosomes, their male and female both are affected. Here the father is affected and mother is the normal. So here from father, daughter and son both are affected. That is, male and females are equally affected. That means autosomal dominant. Dominant. So first you can identify here the generation is skipped or not. If the generation is do not skipped, then dominant. Then you can identify autosomal or recessive. In case of recessive, you can simply find out that mostly males are affected. Okay, but in case of autosomal disease, both male and female both are affected. Here the right answer is autosomal dominant disorder. Next question is, if two persons with AB blood group marry and have sufficiently large number of children, these children could be classified as A blood group, AB blood group, B blood group, 1:2:1 ratio. Modern technique of protein electrophoresis level presence of both A and B type protein is AB blood group of individuals, because here AB blood group means here A and B both are dominant. Both are dominant. In this situation, it's called codominance. Codominance means when both the alleles are dominant. Here, right answer. Next question is, if which one of the which one is the incorrect statement with regard the importance of pedigree analysis. So what is the function of pedigree analysis? Pedigree analysis is a process. Pedigree analysis is a process where the genetist search 10 to 15 or more generation and making a probability the next generation is affected or not in any genetic disorder. Okay? So it confirms the DNA. Pedigree analysis, what is the function of pedigree analysis? The main function of pedigree analysis is to identify the genetic disorder. Okay, the function of pedigree analysis is identify the genetic disorder. Will appear or not in the next generation. This is the function of pedigree analysis. So it confirms that DNA is the carrier of genetic, definitely, and genetic changes occur in DNA. It confirms DNA is the genetic material. It confirms the incorrect statement, not. It confirms that DNA is the carrier of genetic information. It it is right, but pedigree analysis do not confirm it. So wrong. It helps to understand whether the trait is question is dominant. Definitely, here we say before autosomal dominant, X recessive, this type of terms are come from pedigree analysis. So it helps to understand whether the trait is dominant recessive. It confirms that trait is linked to one of the autosome. Definitely it is autosomal dominant, autosomal disease or X-linked disease. They confirm. C right answer. It helps to trace the inheritance specifically. Definitely, a genetist in pedigree analysis, genetist study 10 to 15 or more generation. Okay, to finalize the next generation is affected or not. What is the probability that next generation affected that genetic disease or not? So here the incorrect option is A. Next question is, which one of the following condition correctly describe the manner of determining sex? Sex determination process. Homozygous sex chromosome determine female sex in birth. In case of aves, in case of birds, ZZ and ZW system will apply. ZZ. ZZ represent male and ZW represent female. Female. So this is the wrong answer. Exotype of sex chromosome determine male sex in grasshopper. In case of grasshopper, in case of grasshopper, X0 type of sex chromosome determine the male sex. Okay, this is right. Here, in case of insects, in case of insects, XX and X0 system. XX female represent female and X0 represent male. XO condition in human is found in Turner syndrome. Turner syndrome occur in female, okay, where the 45 one X chromosome is deleted. So X0 means 45 chromosomes are occur. Okay, this is right answer, right option. This is also right. Homozygous sex chromosomes produce male in Drosophila. Right. This is wrong. Exo condition in human found in Turner syndrome determine not determine female sex. It's genetic disorder when the one chromosome is lost. Okay, not determine female sex, because in case of human, sex determination is occurred by SRY genes that present in XY chromosome. Okay, homozygous sex chromosomes produce male, does not. Homozygous produce female. The right answer is B. Next question. Select the incorrect statement from the following. Galactosemia is an inborn error. Galactosemia is an inborn error of metabolism. Here incorrect statement. This is the right option. Small population size. Small population size results in random genetic drift in a population. It is also right answer, because population, when the what is genetic drift? Genetic drift means the change the frequency of a allele in a population. So when number of population decrease in a number of individual decrease in a population, the rate of genetic drift or rate of mutation or rate of evolution is increased. Baldness is a sex determination trait. Okay? Here says baldness is the sex, no, baldness is sex limited. It is the autosomal trait. Autosomal. In case of baldness, the three type of genotype are produced. That is capital B, capital B, capital B, and small b, small b. They are bald. Okay, they are not bald. Linkage is an exception to the principle. So here the incorrect option is C. Linkage is an exception to the principle of independence. Definitely, because Mendel do not describe linkage. Okay, T.H. Morgan first describe this linkage. Okay, and give chromosomal theory of inheritance. This is the right answer. So here the incorrect option is C. Next question is, the genetic defect adenosine deaminase deficiency may be cured permanently by. What is adenosine deaminase? Okay, here we see bone marrow that produce lymphocyte, lymphocyte by adenosine deaminase enzyme. Okay, and lymphocyte produce antibody, different type of antibody that increase our immune system. In case of these genes is mutated, then our body is produced, so no antibody is produced, so SCID disease are produced. It is severe combined immunodeficiency. So here administering adenosine deaminase activator, the genetic defect of adenosine deaminase deficiency may be cured. Okay, introducing bone marrow cell producing ADA into cells at early embryonic stage, enzyme replacement therapy, periodic infusion. This is a wrong answer. This is also wrong. This is also wrong, but this is right. Okay, when adenosine deaminase, it is called gene therapy. Through gene therapy, if the adenosine gene become therapy defected, the adenosine gene is replaced by a good gene. This good gene is called the good gene by the technique is called gene therapy. Next question is A and B genes are linked. Okay, means that it is the chromosome, single chromosome, and single chromosome A and B genes present, but they are closely present. Okay, so they show linked. What shall be genotype of progeny in cross between AB AB and AB AB? So here we can explain properly. This is two chromosome. This is two chromosome are crossed. Okay, here A B and AB, here AB are the linkage means no crossing over is occur. So here two gametes are produced, that is one contain A and B and another contain A B. Here two gametes are produced, one carry A B and another carry A B. If crosses occur but no crossing over, then what happen? One gene carry A B and other genes carry other trait carry A B A B. Okay, these are 100% parental. This is called complete linkage. So here the right answer is AABB. Next question is, in Drosophila, the XXY condition leads to femaleness. Okay, because in case of Drosophila, the sex linked genes are present on the X chromosome, whereas in human being the same condition leads to Klinefelter syndrome. Okay, in male, it proves in human being Y chromosome is active in sex germination. Y chromosome is active in sex germination in both human and being, not false. In Drosophila, Y chromosome decides femaleness, not false. Y chromosome of man have genes for syndrome. No. Here the one copy of X chromosome is increased. So in human being, Y chromosome is active. In human being, this is the Y chromosome, large arm, small arm. In small arm, S or Y gene is present. This sex determination region Y determine the sex in male. Next question is, Betson was the terms used the terms coupling and repulsion for linkage and crossing over. Name the correct parental coupling type along with its crossover and repulsion. Okay, if suppose this is when the A AA and BB present, this is called coupling. This is a coupling reaction. When the genes are close together, that is called coupling. And repulsion means when A B A B, etc. This type repulsion means A B and B. This is called repulsion transform. So coupling means ABB when the same type of genes present in the same chromosome, and repulsion is AABB and this is D answer. This is the 33 D answer. This is coupling means same type of genes are present on the same chromosome, but here different chromosome. This is called repulsion. Next question is, two linked genes and B shows 20% recombination. Okay. Recombination, if we shall go show gametes is 40%. This is C answer. How it is possible? Here we look. Parents generation plus 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 and F1 generation show, and this is AB. Okay. So here, if cross, then this genotype is produced. Now, as given that and the genes show 20% recombination. Okay, 20% recombination means when plus and when plus A or plus B. This is recombination, but this is not recombination. So this is not recombination. So here you can see here this is non-recombinant because the same type of genes present on the same chromosome, but here this is recombination occur. So here only 10%, 20% recombination occur, and 80%, 20% non-recombinant, and recombination occur even, and 80% are non-recombinant. I think you can understand the option. Next question is, of both normal parent, the chance of male child becoming color blind are. So when the color blind are produced, when the first option is the when the color blind, if the mother is at least carrier. Suppose this is mother and this is father. Okay? So when the child become color blind, normal parent, the child color blind, when at least one of his mother, or at least one of his mother is color blind condition, and the mother father color blind, or if mother mother and father both color blind, then child becomes child color blind is a autosomal X-linked recessive disorder. X-linked recessive disorder. So when X-linked recessive disorder means possibly only when all the four grandparents had normal vision. This is not right answer. Possibly only when father's mother was colorblind. Possibly only when mother's father was colorblind. If mother's father is colorblind, suppose X XX and XY. Mother's father is if colorblind, then what happened? This X chromosome go to the daughter, so daughter become carrier. Then if daughter is married a normal man, then when this X chromosome son come to the son are affected. Okay, so either possibly only when mother's father was colorblind, or possibly only when father's mother was colorblind. Father's mother. So father's mother means if father's mother is color blind, and then the father is also color blind, and okay, so then what happened? If normal is mother, father, this is father, this is son, the father's mother, careful, says father's mother, the father's mother is colorblind, then the father is colorblind. If father is colorblind, this X chromosome provide to daughter, the son is not affected. Okay, because this X chromosome go to the daughter, the father provide his X chromosome to the daughter, so daughter become carrier, and son are not affected. But if mother's father is affected, father affected, then what happens? Son, mother, this X chromosome go to the daughter, then from daughter to son, crisscross inheritance. Okay, so here is the D is the correct option. I hope you can enjoy this video properly. If you like it, please like and subscribe my channel. Thank you.